by this presynaptic cell. Same events are going here. But if it is the target cell, like the end, for example, the muscle, so muscle will be excited. So first muscle will get excited, and then it will open other channels through which the sodium will enter, and it will go step by step for the contraction of the muscle. So since we do not have the muscle chapter here, which we finished before, so we are not going to in details how the muscle contraction, but we'll talk about how the signals are transmitted later. So to wrap up the subject of synapse, we have known already that there are presynaptic and postsynaptic cells, and the synapse itself is a cleft, narrow space which separate the post and presynaptic membrane. Okay, and just think about little critically what will be the postsynaptic cells part, and that will be the dendrites. So dendrites of the postsynaptic cell have synapsing with the axon terminals of the presynaptic cells, and the dendrites has those the membrane of the dendrites. They have this receptors proteins, and that will receive the neurotransmitters. Okay, now we see what are the types of synapse. Synapse with another. So this is we are talking about the typing of the synapse based on what we have in the postsynaptic cell. So if the postsynaptic cell and the presynaptic cell both are neurons, then it is a neural synapse, neuron to neuron communications. And if the neuron is synapting with the muscle, then it is called the neuromuscular junction, neuron to muscle communication. And then third type is the gland. So where the it's called the neuroglandular synapses, and it will activate the gland to release the hormones, or if it is other glands, endocrine gland and exocrine glands. Endocrine glands makes the hormone, exocrine makes the other things. Sometimes these axons, what you saw this here, there are multiple. Not well, here you see only one, but many examples are there that this axons becomes branched out. Okay, and then you have another synaptic junctions here. So so there are many of them. So why we have those? Because when we need more communications, more stronger signals, then we need this branching, and this is called the collateral. Collateral branches, side branches of the axons that allow neurons to communicate with several other cells. So we then at the same time we can communicate with not one cell, the target cell, but many cells, and as a result, the communication will be stronger. So here the picture is showing the three different types of synapses. The first one on the top is one neuron to second neuron, and then the middle one is showing you the muscle. The right side is the muscle here. This is the muscle, and it is the neuromuscular junctions. And the third one is the neuroglandular synapses, the gland cells. But all of them are communicating in the same manner. Okay, now neurons. So neurons, going back to the neurons properties. Neurons are the cells, unlike other regular cells like epithelial cells, connective cells, even muscle cells, they do not divide. In most cases, they do not divide. they do not have, they have nucleus, but still they do not divide. One of the reason is they do not have the centrioles. As you go back to the mechanism of mitosis, you need a centrioles to start to begin with the mitosis, and they do not have, and that is the one reason. The other reason is it is a little bit more, I mean, should not say complicated, but you have to remember the cell cycle, which must have been told in either any of the biological class or probably in the AP1. So cell cycle, every cells undergo cell cycle every moment. So these neurons, most of the neurons, they are stuck in the cell cycle, and it is called the G0. The cell cycle usually have Z1, Z2, S phase, 3 phases, and then it additionally Z0 phases when the cell does not do anything. Okay, they do not have to go to the phase of cell division. So these neurons are stuck in the Z0 phase. Just to give you an idea, so if this is the cell cycle, okay, and it is divided into actually mainly three areas or 3 zones. So this one is called Z1. Z1 means growth. This is called Z2. Okay, and this is called the S phase. So each cell goes in around, and then it is actually going from one cell. And then when it finishes the S1, then it goes to the mitosis. So mitosis is very a fraction of the time of the cell cycles. So if you consider this 24 hours for some cells, not every cell is divided in 24 hours. Then it is actually about 1 hour or 1 to 2 hours only for the mitosis. So these cells, these neurons, they've complete G1, G2, S, and then they go, instead of mitosis, they go to another area, which is called Z0, and they stop there. And that's why they do not participate in the mitosis, so they do not divide. So in general, what we learned that neurons cannot divide into daughter cells, and they do not. Only there are some exceptions. Okay, before the exceptions. So if you have a disease or injury, then it is replaced by the new cells coming from the stem cells, and the stem cells will produce the new cells. But in most cases, what happens if, like epithelial cell, if you're injured, the cells will start dividing and make the new cells here, but not here. But there are exceptions of the cell divisions of the neurons. So the exceptions are in the retina. We have neurons. We have in the special part of the brain called hippocampus, which is the area who are involved in the memory function of the brain. And also in the epithelium of the sense of smell, olfactory area, there are the neurons there. These 3 types of neurons, they actually regularly divide. So that these are the exceptions. All right, so the time for first review questions. So we can start discussing this. So name the structural components of the typical neuron. So I said that if you are explaining minimum number of a neuron, that it should be 3 things minimum: the cell body, dendrites, and the axon. But in addition to that, you need to know other part of the neurons, and as well as what are the names, like the rough endoplasmic reticulum is called here Nissl bodies. Then the membrane, plasma membrane, is called in the axon as the axolemma. So this way, the names are bit different from the regular other cells. And when you are describing a synapse, you have to mention that it is the gap or cleft between the pre and postsynaptic cells, and where the release of the neurons are taken, neurotransmitters are taken place. And based on the type of the postsynaptic cell, synapse could be neuron to neuron, the neuromuscular and glandular. And also, if there is no gap, that synapse will be called the electrical synapse. And these are what I just described. These are all chemical synapse. And then finally, why is CNS neuron not usually replaced after it is injured? It is replaced, but it is the word is not correct here. It's why it is not dividing with the new cells. Instead of it is replaced by its new cells, new neurons made by the stem cells, not the original cells which can divide. That is the answer of these review questions. Okay, so now let us now go some easy information for you that you do not have to go in deep thought to understand. So here we are talking about the structural classification. So neurons are the one which perform the functions of the nervous system, carrying the creating the signals, going to sending the signals back to the target cells, and these all are done by different classes of neurons, but most of them are the multipolar neuron, which I showed you the picture. But in addition to that, we have number one, anaxonic neuron, then you have bipolar neurons, and then you have unipolar neuron, and then you have multipolar neurons. What you see here, all those the pictures of all of them. So first, so here, just think about if you compare with the neuron with the glial cells, which will come back later. Glial cells have classification based on their functions. Okay, although they have different structure, or their anatomical structure different, but they are classified based on function. But here they are based on the classification is based on the structure. So first, the left one, all the way here, this is called anaxonic neuron. Anaxonic neuron means what? No axon present. Because when you look at this, everything looks like dendrites, but actually not. In reality, one of them will be actually the axon, and they are compared to others. They are in size. They are very confined. They are little, small in size, and as I said, they have no distinguishing features between the axon and the dendrites, and they are located in the brain and in the special sense organs, and we still not understand all the functions of this. So it is still under investigations. So you see that they are found only in the brain, nowhere else. Okay, next one is the bipolar. So this is the bipolar one. Why it is called bipolar? Because the cell is in the middle, and the dendrites in one side, and axon is in another side. So this is bipolar, and then these are also found in very special sense organs, like eye. We have it. So, but it is also rare. It's not high number. And also, it is found that the maximum maximum length it could have only 30 micrometer. Micrometer is 1,000 of a millimeter, and one millimeter is one small line in your regular, it's called the ruler, measuring ruler. So you see this in the centimeter scale. you'll find one single line small, that's 1 millimeter. And if you divide it into 1,000, one of them is 1 micrometer. So which is not, we cannot see with eye, but we can see with microscope. So that is the size of a maximum size of a bipolar neurons. Okay, the third is the unipolar. Unipolar means you have axons and dendrites at the end. So this is axon. So axon from here to here is axons. And the cell is on one side. That's why it's called unipolar. Okay, and then we have the dendrites here and axon terminals at the other end. So it is dendrites, the cell body lies on the, it is on the one side of the whole structure. Most sensory neurons are this type of. So this is found only in this unipolar one, this number 3 unipolar one, that is found in the peripheral nervous system, because the sensory nerves, which is taking the signals from the peripheral to the central nervous system, these are the one. So this is number is not small, but higher number. And finally, this is the multipolar one. Multipolar is the one which is majority of our neurons are multipolar neurons, and they have why it is called they have called the multipolar, because they have more dendrites than other cells, and with the single axons, and they are highly concentrated in the central nervous system, and they are also motor neurons. Okay, so so this is the sensory, the unipolar, and multipolar are the motor, which takes the signals. So that is the example of the 4 types of neurons. Now, functionally, central nervous system is divided into, again, 3 types. Sensory neurons, motor neurons, we already know, and then we have the interneurons. So what is the interneuron? Interneurons are also the nervous neurons cells sitting in between the sensory and the motor. So now you cannot imagine where we can find the interneurons. Interneurons are found in the brain, and there are 20 billion present in our brain and the spinal cord. Compared to that, we have sensory neurons are 10 million, and motor neurons are 500,000 found in the body. Okay, next is let us see what are the sensory neurons, and these 3 neurons are different or similar in their functions. So in the sensory neurons, it will have the sensory receptors, because these receptors are the one which will detect or receive the signal or stimulations. And based on different types of signals they receive, the sensory neurons are all unipolar, but they are divided on the basis of the receptors. All cells. Okay, so receptor means, of course, these cells, the unipolar cells, but these cells are different on the basis of what type of signal they will receive. So one signal will not be received by the other type of neurons. So we have interoceptors, which are the one present inside the body. Intero means inside. Monitored various different changes occurring in the internal visceral organ. And then from there, the signal will be taken from the inside to the central nervous system. Then sensory receptor also found as a proprioceptor. Proprioceptors are the one which will be present mostly in the joints, and that joints of the body, because they are joints of the body like joints of the bones. Okay. Mainly we have joints between the bones where it is able to move, and when we change our position of the movement, or even we are not going from one place to another place, or we are not moving our body parts from one position and another position, but still the position like sitting, standing, anything, gazing to the sky or anywhere, so your muscles are working, and this, for that, muscles need signals, and these signals are coming from the proprioceptors. And exteroceptors are the one which are present rest of the places around the surface of the body, and then it is the one which will receive the signal from outside. So it includes but not limited to all the general senses, like when you feel hot or temperature changes, when you feel the pressure from outside, when you feel the pain, also you feel the light. Those are the exteroceptors, because they are these receptors, or the neurons are present in the external part of the body. So this is the types of sensory receptors. Sensory receptors are the one which are a member of the sensory neurons, and sensory neurons are, we saw mostly are unipolar. So we can connect all those classification, that classification to these classifications. All right. So next we talk about the interneurons. So sensory neurons goes from the peripheral to the central nervous system. And in the central nervous system, we have a specialized cell which will assess the signals and then decide the new command and then send it back to the motor neurons. So these cells are called the interneurons. Interneurons are located in the brain or in the spinal cord, and they are sitting between the motor and the sensory neurons. And of course, it will receive the signals from the PNS, and also itself, interneurons are member of the central nervous system and also responsible for higher functions. So not only that you need to do the sense and motor action, but some specialized functions which are done by the interneurons, including the memory, learning, planning, emotion. Those you do not need to get reactions or stimulations coming from outside or coming from the inside. It is done, created, and done with the brain itself. Okay, then that third type, which is the motor neurons. These are mainly taking the signals from the central nervous system, and then it is transmitted to the target cells. And the cell bodies are present in the motor neurons cell bodies. Okay, they are, remember, they are the most called the higher. They are the multipolar neurons, and they have the cell, and this cell is present, or cell body or soma is present in the central nervous system, and from there it is extended, their axons in the peripheral system. And if you have the somatic motor neurons, that will go to the skeletal muscles, and it is working under the conscious control or conscious process or voluntary process. And motor neurons, it goes to the visceral internal organ, that these are autonomic nervous system that we have discussed in the beginning, that motor neurons can be two types, or signals could be two types. One goes to the muscle, skeletal muscle, those are called the somatic, and then the other goes inside, which are visceral. And visceral motors, they synapse with the second set of neurons at the peripheral autonomic ganglia. So the synapsing, based on synapsing, there are second synapse also taking place in the visceral motor neurons pathways. But for the somatic, you don't need. The signal comes directly from the central nervous system and goes to the motor or the somatic, and then it is other end is located in the muscles. And so this is the afferent and one when the signals goes from the peripheral to the central. But when we have the signals coming from the central to the peripheral, it is also, this is also called the motor, but they are also known as the efferent fibers or efferent cells. So here is the summary of the cells, of the activities of the different types of nerves, and then how they are connected to the central nervous system and the peripheral nervous system. So if you go from the point where it says stars, so here we have the sensory receptors, either it is internal or external, or it is present in the joints for the positional. Positional and extra receptors both are like outside the body. Outside means not environmental outside, but it is not internal. That's why they are put together, and the interoreceptor means inside the body in the visceral organs. So signals will be received from them, and they will get the signals to the here it will visceral sensory neurons and the somatic sensory. Actually, these are the position is this will go here and this will go here. I'll fix it later. Okay, so this is they all sensory ganglia present in the sensory neurons, and they will get the signals go to the interneurons in the brain. So this is now, so this is the origin of the stimulation area. This is the propagation of the signals area, and this is the central nervous system area where the finally the signal goes to, and then they are analyzed and then go back, sent back to the either visceral motor or the somatic neurons. Somatic neurons goes to the muscle or skeletal muscles, and visceral will go through the, so you have another, you see that directly, the somatic is direct, one single neurons will have, or only one synapse here. But here we have one synapse here and one synapse all the way to the target cell. That is the visceral motor neurons. So in the visceral reactions, again, we have learned that there are two ways, two types of signals. One will stimulate, another will de-stimulate or inhibit the activities of these smooth muscle cells, gland cells. This is the cardiac muscle cells, this is the fat cells. So these are the one activities of this different types of neurons. So we have a review. Classify the neuron according to their structure. So we have 4 types: anaxonic, unipolar, bipolar, and multipolar. And also if you have a questions like to answer, short answer, just do not stop writing there. You have to also, to get the full point, you need to elaborate the answer. In your final exam, you will have many questions which you will need to answer. So if you do not answer all the information which are discussed here, so you will not get the full point. All right, so in this case, you will need to say their locations also. Okay, and then classification of the neurons according to their functions. We have the 3 types. The one which is taking the signal, sensory, motor, and the interneurons. Number 3 are unipolar neurons in a tissue sample Unipolar means the one where the cell is on the side, and they're mostly the sensory neurons. So the questions asking what are unipolar neurons in a tissue sample of the peripheral nerves are most likely to be sensory neurons or motor neurons. This is like a multiple choice questions. So your answer is already showing here with the red, which is a sensory one. Okay, next we talk about the other type of neurons. Sorry, not neurons, the nerve cells. That's called the neuroglia, and simply it is called the glial cells. So why they are important? Because although they are not doing any functional work for the nervous system's function, but they do some other important functions, like they support and protect the neurons. And they are found both central nervous system and peripheral nervous system. Okay, how they support and protect when we talk about each of them one by one, then you will know so what we have, what they are doing. And it is said we find them both in the central and in the peripheral nervous system. But total, we have about six neuroglial cells. And out of 6, 4 are found in the central nervous system, and 2 are found in the peripheral nervous system. Why? Because in the brain, we need some specialized protections and functions that we'll know a little later. They are abundant and diverse. That means that they are very, very high number. Each neuron will have 10 neuroglia surrounding it. So it is the one which is making the half of the volume of the nervous system. So now we see first the 4 types. So let us have the picture, then it will be. If you look at this picture, we have, this is actually showing you a portion from the, what is that? This is the spinal cord. The section by transverse section. So if you have the spinal cord like this, the section is made in this way. And then you are looking from the top or from the bottom of this slice. So now you see that we have learned it before. This spinal cord section, we have a butterfly-like structure that is actually called the gray matter. Gray matter. And gray matter is the one, mostly the neurons, okay, neurons, axons are present, and white matter, sorry, not axon, the cell body are present. And then rest of the area, what you see here, this area, this is called the white matter. And white matters are the one where we have the axons with the myelin sheath. And that's why it is actually like a yellowish, white color area. Okay, so what we are learning here, we are learning what are the different types of neuroglia cells present in the central nervous system. So this is one of them, okay? And these are called the astrocytes. And then we have another type of cells here. We need to know 4 of them. And then this is called the ependymal cells. What else? We have another cells here that's called microglia. Okay? Microglia. And we are left with only one cell, which is this one here. This is called oligodendrocytes. So oligodendrocyte 1, 2, 3, and 4. These are the 4 cells, 4 neuroglial cells present in one neurons. Present in the central nervous system. Now, you also notice that this is one neuron, multiple neuron cells is present in the brain or spinal cord. And it is also showing you the how they are present. You see all the cell bodies are in the gray matter, and all the white area or white gray materials and white materials of the spinal cord, they are present in the periphery or the white areas. So now what they do, these 4 cells. So first we start with the what? Ependymal cells? No. No. Let's see what we have. Yeah. Okay, here we have number 1 is ependymal cells. Ependymal cells, I have shown you in the picture. I'll show you later. So what they do, they form simple cuboidal to columnar epithelial cells called ependymal cells. Okay, now these are the cells. They are lined fluid filled passageway, and that is called the ventricles. Ventricles, in your lab, I explained to it. In the brain, we have 1, 2, 3, 4, total 4 ventricles. And ventricles are like open space where we have fluid. The fluid is called the cerebrospinal fluid. This cerebrospinal fluid actually are made by the ependymal cells. And also it is the one not only producing, circulating and monitoring this central nervous system. So cerebrospinal fluid is the main functions. So why we need cerebrospinal fluid in the brain? Because first of all, it is protecting, physical protection and nutritional protection. Nutritionally, it is supplying the food to the cells in the brain. So look at this picture again. Where is the ependymal cells? Here, these are the ependymal cells, okay, like pink color. And they are discharging the fluid here. This is the cerebrospinal CSP. And this is actually another ventricle called the lateral ventricle. All right, next one is the astrocytes. Let me see what we have. Microglia. Microglia are the cells which are monocytes. Monocytes are white blood cells. Macrophages are the like differentiated white blood cells, which are the monocytes. Monocytes and macrophages. Monocytes are not differentiated, but macrophages differentiate. That means they become very big, and both of them are the one who are they are capable of killing the germs. So they migrate into the central nervous system during the nervous system formation, but originally they were white blood cells, and they persist as mobile phagocytic cells, and they remove the cellular debris, waste products, and pathogens. So this is where our neurons in the brain and spinal cord are protected and remain as the active one. The third type of cells are astrocytes. Astrocytes are performing very, very important function, which is called the blood-brain barrier. So how it is done, blood-brain barrier. So you go back to the picture again. So what you see here, these are the astrocytes, this one. And you see they are sitting and holding in one side the axon of the of the neurons, and other side it is holding the blood vessels. This is the blood vessels, okay? So now any germs will try to come out, okay, and affect from the blood, because blood is the one which carry the anything from one part of the body to the other part. So it can also go to the brain. So these germs will come out and affect the neurons. So that it doesn't happen, these cells are making a barrier between this blood and the neurons. That's why it is called the blood-brain barrier. So that is made by the performed by the astrocytes. Okay, then we have the oligodendrocyte cells. This is the one. Yes, astrocytes. So they also absorb and recycle their neurotransmitters, and they form the scar tissues after CNS injury. These are other functions of the astrocytes. So next one is oligodendrocyte, number 4, and that will be the last one for the central nervous system. And they are the one who provides central nervous system framework by stabilizing the axons. How? They actually grab the axons with the fat materials, which is called the myelin or myelin sheath. So myelin sheath is an additional cover in that is present in the axon of the neurons, and that is made by the oligodendrocyte cells in the neurons of the central nervous system. So if you look at the picture again, oligodendrocyte cells are this one here, and you see that it is covering with the myelin sheath. So myelin sheath is a protecting cover, and its materials are fat around the neuron, around the axons of the neuron. And when we have this myelin sheath, the signals will go very fast from the cell to the axon to the terminal. And if it doesn't present, then it will go slow, okay, slowly. So when we need faster propagation of the signals, then we must have wrapping of the axons with the myelinated sheath or myelin sheath that is produced by these cells called oligodendrocyte cells. We have also similar cells in the peripheral nervous system doing exactly the same functions, but they have different names. So the rest of the 2 of the 6 glial cells we'll talk about after we finish this part with the review questions. Okay, so read this review question. First of all, identify the neuroglia of the central nervous system. So if you get this question in your final exam, you do not just write the names of the 4, which are astrocyte, oligodendrocyte cells, then what else? Ependymal cells and neuroglia. Astrocytes, oligodendrocytes, and what is the other one? Astrocytes and microglia.
Are you saying don't just list them? You want us to?
I said no, don't just write down. So also you need to write what they do, on the function of the 4. Number B, which glial cells protects the CNS from chemical and hormone circulating in the blood? It will be the what? Astrocytes. Okay, and for these questions, you should need also to explain how it is protecting. It is making the blood-brain barrier. Number C, which of type of neuroglia would increase in the brain tissue of a person with CNS infections? And that will be the microglia. Did you understand what the question is here? So when we have infections,
Yeah.
the cells who protect us from the infection, their number increases. So this is true for our regular infections in the blood. So the blood count of the white blood cells goes high because they are fighting and they make more cells. You need more soldiers to fight against the germs, and that's why their number increases. And again, what type of infections you have, either it's a viral infection, bacterial infection, or like the fungus infections, based on that, or inflammation or the chemicals, the different types of white blood cells will increase. But that is not the case in terms of the nervous system. All right. So next we'll have the peripheral nervous system. And peripheral nervous system have only two types. One is called the Schwann cells, and the other one is called the satellite cells. Schwann cells are the same as the ependymal cells in the PNS. They make the myelination over the axons, and this because of this myelination, the axons are capable of transmitting the signals in high speed. Without myelination, the speed will be very low. And then we have the satellite cells, which is surrounded peripheral cell bodies, and it is the one which regulate the environment, and it is same function as the astrocytes. Astrocyte cells are the one which is making the blood-brain barrier, but here it is not that much functions like the astrocytes, but they help the neurons to not to get infections. Also, another functions done by the satellite cells, if there are injury, they are the one which helps to make new cells from the stem cells. That's the satellite cells. All right. So now just we have found that if the axons are coated with the myelin sheath, it will be protected first from being injured or from being damaged, as well as the signal will go faster. So how they are actually found, see here, this is a Schwann cell. That means it is in a neuron found in the peripheral nervous system. And Schwann cells is wrapping around the axons, but the wrapping is not continuous. It is one cell, then you have a gap, and then you have another cell, then you have gap. So the gap is very important, and we'll discuss that later also, why we need a gap. So this is the myelinated sheet made by the Schwann cells, and then the one which is not, the portion which is not myelinated in between, these are called the nodes, or and full name is nodes of what is that called? Ranvier, yeah. Okay. And myelinated here, myelin sheath in PNS is formed by the Schwann cells, and myelin sheath in CNS formed by the what? Oligodendrocyte cells. Myelinated axons, outer surface is called the neurolemma, and a single Schwann cells wraps a single internode. So it is not that continuous. And because of this nodes, the propagation of the signals is step by step, not the continuous one. And if the axons are not myelinated, a single Schwann cells can enclose multiple segments, and it stabilizes the position of the axons. And subsequent pictures, you can see how it looks like when we cut the axons with the Schwann cells on it, or the myelin sheath on it. So remember, Schwann cell is the cell, and the cell makes the cover sheathing. That is called the myelin sheath. Myelin sheath is not the cell. It is created by the cells. So now in the peripheral nervous system, the neuroglia also, as I said, they help to get the repair done. And this is one information in details of how the repair process takes place in the PNS. First, the axon and myelin degenerates, which is far away from the injury. So if an injury is here, the axon will degenerate in the quite distance, because when the repair starts, it will start from that places. And then the cells which are making the myelin sheath will proliferate in the line of its axon's length. And in this case, in the peripheral nervous system, we have white blood cells called macrophages. It's not the astrocytes here. Astrocytes will do the same thing in the central nervous system. But here, the macrophages move in this area, and they will eat up the debris and eat up any germs there. An axon grows along the path created by the Schwann cells. And Schwann cells then wrap around the elongated axons. And if normal synaptic contacts reestablished, normal function may be regained. If an axon stops growing or grows in wrong direction, normal function will not return. So that means we can have a possibility of repair in both directions. And this repair process, whole process, is called the Wallerian degeneration until the function is restored. So here, with the cartoon, we can explain how the repair process takes place. So you see here, the injury took place, first picture, and then it is distal to the injury side, the axon and myelin sheath degenerates. Okay, so this is not the injury, but this is the like injury means that self-sustained injury takes place, site of injury, and then we have a so axons and myelin sheath degenerates in the distal area. And step 2, Schwann cells proliferate, and that means they make proliferation means making many of them. Okay. Like not 1 to 2, 2 to 4, but many of them will just will make here. Schwann cells proliferate along the original axon path. And number 4, Schwann cells wrap around. So you can see this one here, this wrapping around, and axon grows into injury sites along with the Schwann cells. So it is going further down, so where the original injury took place. So review questions here. Identify the neuroglia of the PNS. We have 2 of them. Okay, Schwann cells and satellite cells. And describe the neurolemma, which is the membrane of the axons with the myelin sheath, and in this case with the Schwann cells. And in which part of the nervous system does Wallerian degeneration to occur? In which part of the nervous system? It is in the peripheral nervous system. And then it is not exactly at the, it doesn't start exactly at the place where it was injured. It is this little distal pathway. Distal means away. So we have a video to show you, and then we'll start the second area. So second and the most difficult part, which is how the signals actually propagate. So I'll not start that today. I will rather show you the video, and then I'll take your attendance, and then you will be done for the day.
The Empire cannot withstand.
So the purpose of showing you this video to get you some information ahead of what I'm going to talk about in the next episode, which is the action potential.
Nervous system is going to be made up of your brain, and your central nervous system is the white matter, parietal lobe, hand, and I didn't work together.
Oh, not this one.
Then we're going to stay in the cerebrum to
Neurons are the cells that make up our nervous system, and they're made up of three main parts. The dendrite, which are little branches off the neuron that receive signals from other neurons. The soma, or cell body, which has all the neuron's main organelles like the nucleus. And the axon, which is intermittently wrapped in fatty myelin. Those dendrites receive signals from other neurons via neurotransmitters, which, when they bind to receptors on the dendrite, act as a chemical signal. That binding opens ion channels that allow charged ions to flow in and out of the cell, converting the chemical signal into an electrical signal. Since a single neuron can have a ton of dendrites receiving input, if the combined effect of multiple dendrites changes the overall charge of the cell enough, then it triggers an action potential, which is an electrical signal that races down the axon up to 100 meters per second, triggering the release of neurotransmitter on the other end, and further relaying the signal. So neurons use neurotransmitters as a signal to communicate with each other, but they use action potentials to propagate that signal within the cell. Some of these neurons can be very long, especially ones that go from the spinal cord to the toes. So the movement of this electrical signal within the cell is super important. But why does a cell have an electrical charge in the first place? Well, it's based on different concentrations of ions on the inside versus the outside of the cell. Generally speaking, there are more Na+ or sodium ions, Cl- or chloride ions, and Ca2+ or calcium ions on the outside of the cell, and more K plus or potassium ion, and A minus, which we just use for negatively charged anions, on the inside of the cell. Overall, the distribution of these ions gives the cell a net negative charge of close to negative 65 millivolts relative to the outside environment. And this is the neuron's resting membrane potential. When a neurotransmitter binds to a receptor on the dendrite, a ligand-gated ion channel opens up to allow certain ions to flow in, depending on the channel. Ligand-gated literally means that the gate responds to a ligand, which in this case is a neurotransmitter. So let's take the example of a ligand-gated sodium ion channel, which when it opens lets sodium flow into the cell. The extra positive charge that flows in makes the cell less negative, since remember it's usually negative 65 millivolts, and therefore less polar. So that's why gaining positive charge is called depolarization. Neurotransmitters typically open various ligand-gated ion channels all at once, so ions like sodium and calcium might flow in, while other ions like potassium might flow out, which would actually mean some positive charge leaves the cell. In the end, though, when it's all added up, if there's a net influx of positive charge, then it's called an excitatory postsynaptic potential, or EPSP. In contrast, the opening of only ligand-gated chloride ion channels would cause a net influx of negative charge, creating an inhibitory postsynaptic potential, or IPSP, making the cell potential more negative or repolarizing it. Now, a single EPSP or IPSP causes only a small change on the resting membrane potential. But if there are enough EPSPs across multiple sites on the dendrites, then collectively they can push the membrane potential to a specific threshold value, typically around negative 55 millivolts, although this can vary depending on the tissue. When it hits this threshold value, it triggers the opening of voltage-gated sodium channels at the start of the axon, which is called the axon hillock. These voltage-gated channels open in response to a change in voltage, and when these open, sodium rushes into the cell. The influx of sodium ions and the resulting change in membrane potential causes nearby voltage-gated sodium channels to open as well, setting off this chain reaction that continues down the entire length of the axon, which ends up being our action potential. And when this happens, we say that the neuron has fired. Once a lot of sodium has rushed across the neuronal membrane, the cell actually becomes positively charged relative to the external environment, up to about positive 40 millivolts. This depolarization process ends when the sodium channel stops allowing sodium to flow into the cells, which is a process called inactivation. But this inactivated state's different from when the channel's closed, or open for that matter, which are the two states that most other channels have. The voltage-gated sodium channel, though, is unique in that it has what's known as the inactivation gate, which blocks sodium influx shortly after depolarization, and stays in this state until the cell repolarizes and the channel enters the closed state again, and the inactivation gate stops blocking influx. Even though that inactivation gate's not blocking it anymore, the channel's still closed, so no sodium enters the cell in this state.
When your kid is hurting and there's nothing you can do about it.
The middle open state's therefore the only state where sodium gets let into the cell through the channel, and this is a very short window of time. Now, in addition to these sodium voltage-gated channels, we've also got potassium voltage-gated channels, which are slow to respond and don't open until the sodium channels have already opened and become inactivated. The result of this is that after that initial sodium rush into the cell, potassium flows out of the cell down its own electrochemical gradient, removing some positive charge and blunting the effect of the sodium depolarization. These potassium channels don't have a separate inactivation gate, and therefore they stay open for slightly longer, which means that there's a period of time when there's net movement of positive ions out of the cell, which causes the membrane potential to become more negative, or repolarize. During this repolarization phase, the cell also relies on the sodium-potassium pump, which is an active transporter that moves 3 sodium ions out of the cell and 2 potassium ions into the cell. It's during this repolarization phase that the cell's in its absolute refractory period, since the sodium channels are inactivated and won't respond to any amount of stimuli. This absolute refractory period keeps the action potentials from happening too close together in time, and also keeps the action potential moving in one direction. The combined efforts of this pump and the extended opening of the potassium channels result in a small period of overcorrection, where the neuron becomes hyperpolarized relative to the resting potential. During this time, the sodium channels go back to their initial closed state, and for a short period, the potassium channels stay open. At this point, we're in the relative refractory period, since the sodium channels are closed, but they can be activated. Although because the potassium channels are still open and we're in a hyperpolarized state, it takes a stronger stimulus to do so. Finally, as the potassium channels close, the neuron returns to its resting membrane potential. All right, as a quick graphical recap of this whole process, with membrane potential on the y and time on the x. First, we start at the resting membrane potential of around negative 65 millivolts, and voltage-gated sodium and potassium channels are closed. We then receive EPSPs enough to hit threshold at about negative 55 millivolts, causing voltage-gated sodium channels to open, and we reach a peak of about positive 40 millivolts, at which point the sodium channels become inactivated, and we're in the absolute refractory period. Voltage-gated potassium channels then open, and along with the sodium-potassium pump, start to repolarize the cell. So much so that it overshoots and hyperpolarizes the cell. Next, the sodium channels enter their closed resting state, while at the same time potassium channels start to close, meaning that we're in the relative refractory period. Until finally they all close and we get to our resting membrane potential once again. Alright, so this process of positive sodium ions moving in and depolarizing the cell transmits the electrical signal down the length of the axon. Great. But really this process isn't that fast. So that's where the fatty myelin comes in, which comes from glial cells like Schwann cells or oligodendrocytes. These myelinated areas don't have voltage-gated ion channels spanning the membrane, so ions can't simply flow into the cell. That only happens in the spots between the myelin, called nodes of Ranvier. So instead of propagating via channels, the charge essentially jumps from one node to the next. That said, though, jumps isn't really the right term, and these ions aren't just diffusing down the length of the myelin to the other side. That'd be way too slow. What actually happens is more like the sodium ions rush in and bump other positive sodium ions already inside the cell, which bumps other ones, and so on until this wave of positivity reaches the next node. The charge moving in this way down the myelinated areas moves really fast and is called saltatory conduction, which makes it look like the action potential jumps from one node to the next. All right, as an extremely quick recap, neuron action potentials happen when dendrites receive enough EPSPs to open voltage-gated sodium channels, which causes rapid depolarization of the neuronal membrane and propagation of an electrical charge from node to node down the length of the axon.
All right, thank you. So let me have your attendance. Yeah. Ashish Shade.
I'm here.
Diamel.
I'm here, Professor.
Dorothy.
Yeah.
Julia. Rowan.
Yes.
Jaden. Marianne.
Here.
Diamond.
Here.
Matthew.
Here.
Noshia.
Here.
Wasi, Chanel, Coseda, I think Coseda, you asked me a question in the beginning. Rainy,
Present.
Angelica, I think she's not here. Noor, Findadigo,
I'm here. Yeah,
thank you. Alsain,
thank you.
Here.
Sante, Rachel, Gundo,
Present.
Alisa,
Here, Professor.
Natalia.
I'm here, Professor.
You are not there yesterday.
Yeah, I wasn't there yesterday because I had an appointment.
Right, so you missed a lot of info.
It's okay. I clicked in with somebody and I got the notes from yesterday.
Okay, have a great day, Professor. Bye.
Bye-bye.
Professor, I think you missed my name.
Bye, Professor.
Yes,